Thanks a lot, Huanqi. Thanks a lot for the invitation. Thank you all for coming and braving the weather. I probably will just spend the night here. Yeah, Huanqi basically gave the whole talk in a nutshell, so I'll just fill in the rest of the details. I'm going to talk to you about the work that I did in Adam Cohen's lab over the last 6 and a bit years on developing molecular and optical tools for all-optical electrophysiology. And then I'll tell you how we're hoping to bring these tools to hopefully everybody in this room here through a new platform that we're setting up at the Broad. And so this is just the picture version of what Huanqi just said. I don't have to extol the virtues of electrophysiology to this crowd. It's one of our most sensitive biological techniques that we have. It goes down to single molecule sensitivity. It lets us look at the input-output properties of neurons. But I also probably don't have to sort of disparage its limitations. This is one of the highest throughput experiments that has been done electrophysiologically. This can record 12 cells. For reference, there are 80 million cells in the mouse brain and 80 billion cells or 80 million neurons in the mouse brain and 100 billion neurons in the human brain. So there's a little bit of a disconnect there. It also takes a long time and is done in a dark, sad room a lot of the time. So we wanted to see if we could do better. And one of the key things that you can do with electrophysiology is it gives you bidirectional control over the cells. That means that you can simultaneously apply stimulation paradigms and then record what the cell does as a response. And so tools have become available over the last decade to do both of these individual tasks using light. For example, many of you know CHArT, which is a blue light-gated ion channel, which was first used by cyanobacteria to make energy. But in 2005, it was discovered that this protein could be expressed in mammalian neurons and used to trigger action potentials on demand by using flashes of blue light. And because you can shape light very easily, both in terms of space and in terms of time, you can project custom patterns of activity into neural tissue. At the same time, people were also developing calcium indicators. These are fluorescent sensors based around modifications of GFP, which modulate their fluorescence as a function of the calcium concentration inside the cell. And because neurons tend to couple calcium influx with electrical activity, you can use these sensors as proxies for neuronal or other cells' activities. And so the question is, can we combine both of these and achieve the bidirectional control that electrophysiology affords you? And so to do that, you want to ideally co-transfect with an actuator and a reporter protein. And you want to do a two-color experiment where you have one color of light, which is used for perturbation, and one color which is used for reading out the resulting activity. And so the trick is you want the perturbation light to have a very sensitive and temporally precise activation of the actuator, but you also want to make sure that it doesn't induce any photo artifacts in the recording channel. You want to make sure that your recording light, on the other hand, gives you very high signal-to-noise recordings of the underlying activity, but you want to make sure that it doesn't cause any aberrant activity through activating the actuator. And so basically my entire PhD was spent figuring out how to make these two elements very big and the other two elements very small. The reason that this was a challenge is that the calcium indicators and the CHArT that I showed you 2 slides ago have a large degree of spectral overlap. So here I'm showing you a spectrum of well, not all of the available, but a sampling of available CHArT actuators. And you see that the peak wavelengths really do span the entire visible range. However, if you now overlay the GCAMP6 absorption spectrum, and GCAMP6 is the best current fluorescent calcium indicator, you see there's no wavelength you can use to image GCAMP which won't also drive every single one of these CHArT with at least 20% activity. And this is a problem because you actually usually need more light to do GCAMP imaging than to do CHArT activation. So you will basically get the cells firing. However, if you look at the red half of the spectrum, there are a lot of wavelengths still available which would be able to avoid activation of the blue-shifted CHArT. And so we set out to think about how we can combine blue-shifted CHArT with red-shifted activity indicators. Well, sorry, that's all the talk we have for today. Here we go. And so redshifted indicators for neuronal activity do exist. On the top, this is actually data that I took of one cell which I patch clamped and then recorded both a calcium response with a redshifted calcium indicator, which was developed. The most recent and best versions are coming out of Genelia Farm from Doug Kim's lab. And also, there are redshifted voltage indicators, which instead of using calcium as a proxy, directly report on the electrical activity of the cell. And those actually came out of Adam's lab right around when I was joining. And so I did this 3 different ways. And I'm not going to talk about all of them. But in each case, we were able to combine a spectrally orthogonal pair of proteins with some sophisticated optics in order to allow bidirectional control of tissues. I'll show you 2 examples. The first one is a little more intellectually similar to the introduction, so I'll start with that, even though we did it second. And the goal we had here was to use a redshifted calcium indicator and blue-shifted channelrhodopsin to record from as many cells as we possibly could inside an intact brain tissue. So like I said, with electrophysiology, that number is 12, and we wanted to get it to the thousands. And before I forget, I need to acknowledge Vicente, who's my co-conspirator and commiserator throughout this whole project. He really drove the optics that I'll present in a little bit. And so the 2 challenges we had to achieve this project were that we had to find a spectrally orthogonal pair of a reporter and actuator. And we also had to figure out how we were going to do the imaging over millimeter scale sections of tissue with single cell resolution. I'll show you how we did both of these things, and then I'll show you very quickly one application of mapping the effect of anti-epileptic drugs throughout acute slices. So to begin with, we went through the currently available redshifted calcium indicators and just co-transfected them in cultured neurons and drove single action potentials to make sure that the literature was right, and it was. The JR-Gecko 1A proved to be an extremely sensitive and fast calcium indicator, exactly as had been found before. This is good because we need a high signal-to-noise recording, but we also want to make sure that we don't have problems with blue light activation causing photo artifacts. And to do that, we transected these proteins in hex cells and just applied pulses of blue light while monitoring the yellow light-induced fluorescence. We found that, as people had suspected, blue light induces photo switching in all 3 of these proteins. But we wanted to be a little bit more careful with our characterization. And we measured the photo switching as a function of blue light intensity, finding that while JR-Camp 1A showed very little photo switching, JR-Gecko 1A and RCamp 2 showed a great degree. Unfortunately, JR-Camp 1A is a pretty bad calcium indicator, so we didn't want to use it for these wide field measurements. Instead, we looked at the photo switching activation curve and saw that this would be great to have a stick, but while typical optogenetic activation is done in this intensity region, if we could take a factor of 10 off the amount of blue light we needed, we could reach a region with no photo switching. And so to do that, we went around looking for a sensitive enough channelrhodopsin to require less blue light for photoactivation. We stumbled upon a protein called CHArT in a screen done by the Boyden lab, which was appealing to us because it's very blue shifted. It's about 50 nanometers blue shifted, or 25 nanometers blue shifted relative to channelrhodopsin 2, which would be great for avoiding spectral crosstalk. But the problem, at least in the original paper, is that the photocurrents reported were very poor. We knew this paper very well, however, and we knew that a lot of the problems with photocurrents in that particular paper were because the proteins hadn't trafficked well to the cell membrane. So we did a very simple trick. We appended a Kerr 2.1 trafficking sequence to CHArT and found beautiful trafficking in cultured neurons all the way out to the dendritic spines. And we found that when measuring the current by whole cell patch clamp, it was actually double that of channelrhodopsin 2 H134R, which is the neuroscientist's standard channelrhodopsin. We also found that not only was TSCHR, not only did it have higher photocurrents, it actually had more sensitive responses to blue light, requiring about a factor of 10 to 100 less light to achieve the same photocurrents that you can get with channelrhodopsin 2. And while the blue light photocurrents are higher and more sensitive, when we measured the yellow light photocurrents, we found that they were low both relative to channelrhodopsin 2 and low in an absolute sense in the single-digit picoamps, which are unlikely to cause any defects when imaging JR gecko simultaneously. So then we co-transfected JR gecko and TSCHR into the same cell. Here I'm showing the TSCHR by its GFP label. And we applied a ramp of blue light indicated on the bottom. These are 10 millisecond pulses while monitoring the fluorescent response of the JR gecko channel. And you see that at a certain threshold intensity, we start to get calcium transients. These calcium transients go away when we block them with TTX, and they come back when we wash them out, suggesting that they are caused by activity of the neuron. The little remaining sharp blips are just direct fluorescence caused by activating the JR gecko by the blue light. And this is basically going to be easily corrected in the remainder of the talk. I wanted to show you raw data first. The problem we had, however, is that when we took these constructs into intact tissue, into slices, the fluorescence of the neuropil signal dominated the somatic responses. To clean that up, we attached a nuclear targeting domain. This is just a section of the H2B protein, which localized the JR gecko to the nucleus and allowed us to get very clean single-cell signals. And what we paid for this is a decrease in the speed, but the protein was still responsive to a number of action potentials in the linear fashion. This was enough to remove the neuropil signal, but it wasn't enough to remove the out-of-plane signal that we get when imaging intact brain slices. When you want to look at live tissue, you usually want to cut 300 micrometer brain slices, and you have many cells both above and below the plane contributing signal. So we wanted to figure out an optical way to get rid of this signal, and we built a custom microscope with a very large field of view. This giant objective is actually pretty much true to size as you see it there. It has a 5 millimeter field of view and a 0.5 NA. These are big numbers, let's put it. And we just modified a standard microscope with this objective with a DMD. A DMD stands for a digital micromirror device. This is basically a projector. And we used this projector to apply patterns of light for JR gecko stimulation, such that adjacent regions of the sample got temporally orthogonal sequences of yellow light, and such that over any particular space in time, the light was on for half the time. This allowed us to be efficient in our use of photons. We then were able to acquire a series of images with different illumination patterns, which is represented over on the left. We were able to use our knowledge of these patterns to demodulate the individual XY locations. Then we were able to apply a synthetic aperture. This was all done in silico after the data has been acquired to remove out-of-focus light. And then finally, we were able to reconstruct optically sectioned images. And to show you what this looks like in real terms, this is now a slice of the mouse thalamus and hypothalamus. This is something we just had in the fridge. This is RBP4 Cree expressed, an RBP4 Cree mouse expressing M-citrine. And what I'm showing you is a Z-stack acquired in wide field where the signal from every plane is encoded using its color, using a color value. And so you can see that although maybe you can see some colors, grayscale really does dominate the signal. If we now do the same thing in Hadamard, you see that we're much better able to localize the Z location of individual neurons, and we're able to see how these fiber tracts loop throughout the slice. We can zoom in on a little region to give you a better view. And you can see that Hadamard imaging effectively removes the out-of-focus contribution from fiber tracts as well as out-of-focus cells. So now that we had a molecular technique combined with an optical technique, we were ready to do a real experiment. And we packaged both constructs into AAV9, co-injected them into neonatal mouse pups. Everything is driven by the synapsin promoter. We're not doing anything to be specific with what we're expressing for. And we developed an imaging and stimulation protocol so that we are stimulating with progressively increasing amounts of blue light. Then, by monitoring the response of the cells as a function of blue light intensity, this will give us a readout of the cell's intrinsic excitability. So basically, how likely is a cell to fire given a certain amount of input? This is what the image, this isn't looking great, but whatever. This is what the CHArT image looks like from one field of view of this microscope. This is the JR gecko signal. And now on the next slide, I'll just show you the change in fluorescence as a result of blue light in red, so you can see that we have activation from many cells. We were able to pull out 6,102 cells out of this particular brain slice, about half and half from cortex and striatum. That just happened to be where our injection site landed. And we're able to see at least qualitative differences between the responses in the cortex and striatum, which is encouraging that we're looking at something that has something to do with biology rather than technical artifacts of our measurement. We were, of course, then able to map the excitability profiles back into XY coordinates. So we did a principal component analysis, basically to generate a pretty color space. The point is that cells which are more pink end up being less excitable. Cells that are more green are more excitable. And brighter cells just had stronger responses. So here I'm showing you 3 traces from cells 1, 2, and 3. And so with that, we wanted to do a quick application to show the value of this tool. And because we were measuring excitability anyway, looking at the action of anti-epileptic drugs seemed like a good place to start. We chose 3 drugs. Carbamazepine and phenytoin are commonly prescribed, but have very unclear mechanisms of actions. Retigabine, on the other hand, is not on the market anymore because apparently it turns people blue. I still don't understand how this happens. But it has a pretty specific mechanism. It's a positive allosteric modulator for Kv 7.2 and 7.3 heteromers. So I'll show you what happens now to the change of responses before and after retigabine application. Here in green, green cells have stronger responses to the drug, meaning they exhibited a greater degree of silencing, whereas cells that are magenta had lower degrees of silencing. And you see there is a spatial profile to the response of this drug's action. We plotted this as a function of depth along the cortex, averaging cells along the way. And then we're curious to see if this matched up with the known expression profiles of KCNQ2 and KCNQ3, which are Kv 7.2 and 7.3. So we took data from the Allen Brain Atlas and mapped it onto the same scale, and we're very gratified to see that cells which showed a large degree of inhibition by retigabine also happened to be in regions which had a large degree of expression of its target, which was gratifying. We were also able to map the profile of carbamazepine, phenytoin, and retigabine on the same scale. This is now 35,000 cells from 9 slices in 3 animals, measured in 7 hours or so. And you can see that the 3 drugs with potentially 3 different mechanisms of action do have different response profiles across the cortical depth. And so this is around where I want to leave you with this tool. This is also where we left the reviewers. But I wanted to just say that this could be a nice phenotypic assay to very quickly figure out where a pharmacological reagent is acting within the brain. We did 9 slices here. It would have been just as easy to do many more. But now I want to think about, what about a more detailed view? So far, everything we've been looking at has been calcium activity, which is low-pass filtered relative to the underlying voltage signal. And so to tell you about that, I'll return to an older project, which is very similar in nature, but a combination of channelrhodopsin and a voltage indicator. This was work that I did together with Daniel Hochbaum and Yongxin Zhao. Yongxin was with Robert Campbell's lab at the time. And the work begins in like 2010 with the discovery that archirhodopsin 3, which is a red or a orange light triggered proton pump, also happens to be very weakly red light fluorescent in 640 nanometers. It also turned out that this fluorescence was voltage dependent. So here we're showing a hex cell expressing archirhodopsin 3. And as the voltage is modulated by a patch clamp electrode, you see very fast responses in the cell's fluorescence. And so this protein is redshifted enough to be combined with channelrhodopsin 2 in principle. And now I'm going to very quickly go through the problems and solutions that we had with this project. So it turned out that imaging ARCH on its own perturbs voltage directly. This is because natively this channel is actually a pump. Oh, I'm sorry, this molecule is actually a pump and is supposed to perturb voltage. We found that exciting the channelrhodopsin causes photo artifacts, the same as the problem we had with JR Gecko. And we found that imaging ARCH on its own can excite channelrhodopsin 2 activity. So we had all of the problems. And we did about 3 years of solutions. We did directed evolution on the ARCH to get rid of the photocurrent and to make it brighter. We found more sensitive channelrhodopsins from the literature. And we performed targeted mutations on them to blue shift their spectrum. So at the end of the day, we had exactly the same cartoon with different protein names, which were now spectrally orthogonal. So now this is what we all came for. We came for the pretty images. This is now a neuron co-expressing Sheriff and Quasar 2, where I'm pulsing flashes of blue light while watching the fluorescence response. And you see that we can track action potentials happening very clearly. We can get a little bit more sophisticated with this. This is now another cultured neuron, which I'm stimulating using a DMD with blue light only in this region. And then we've taken some computational approaches to average together many repeats of many action potentials and interpolate the initiation time of the action potential in every point in this movie. And so I'm going to play it, and then I'm going to tell you what to look for. So hopefully you can see that the action potential is propagating across the entire field of view. And now if you watch a little bit more carefully, you'll see that even though we're stimulating over here, the action potential is initiating over here and then propagating throughout the entire cell. I'm going to let that go one more time so that everybody can see it. There it is. And this was actually hypothesized for a very long time that action potentials should initiate in a part of the cell called the axon initial segment. So what's happening is we're stimulating dendrites and some small process. Potentially the axon is initiating the action potential. But you can perform immunostaining to confirm that the axon initial segment is actually the location by staining for anchorin G, and you see very nice correspondence between the two. We can also perform these measurements at low magnification. So this is a totally different microscope, but the idea is exactly the same. We're measuring now many more cells simultaneously. And I'm showing you the activities of each cell color-coded while stimulating with a low level of blue light. So this is something like 20-some cells. We've now gotten these measurements to go all the way up to 100 neurons simultaneously. And that's actually something that we'll be setting up in-house here. These are cultured neurons, yes. In slice, the most we've gotten simultaneously is like 6 or 7. The problem is that these are still very dim, and if you put more light into the slice, you will burn it. I think right now Vicente is pushing that envelope a little bit. He's getting to the low dozens, but that's still work in progress. And then you sort of preempted me a little bit. These do work in brain slice. These are organotypic brain slices, but we've since gotten them to work both in acute brain slices and most recently, coming any day out now, Yoav's paper on getting this to work in vivo in the hippocampus. So I think there he shows 7 cells monitored simultaneously, 4 of which responded, which potentially was actual biological signal, but I don't know enough about the hippocampus to judge.
So the major advantage of the calcium sensors is just how much less light you can use for those?
So voltage imaging takes about as much light as 2-photon imaging. Both of those limit you to about a 200 micron field of view before putting in too much power into the brain and starting to damage the tissue. Calcium imaging requires 10 to the 4-fold less light, so that's 10 to the 4-fold bigger field of view potentially before running into that particular limitation. So the field of view that we were mostly limited by the size of our viral injection region, which I'm hoping that Ben Deverman fixes for us soon. And then after that, we were limited by the size of our objective, which could only cover a third of a coronal brain slice. So, like I mentioned, this technology has been available for a little bit of time. And I just want to walk you very quickly through some of the applications that people have used, both in our lab and without. So this is a paper that came out earlier this year using Optopatch, or All-Optical Electrophysiology, to monitor the effects of the SOD1A4V mutation in human iPSC-derived motor neurons, finding that it lowered the the excitability at high stimulation regimes. This was nice because it sort of reconciled two apparently contradictory findings in the literature. Some people were making these neurons and finding that the mutation had no effect on the firing rate. Some people were making these neurons and found a lowering of the firing rate. And it turned out that the answer was yes, both. At the low regime, there was no difference. At the high stimulation regime is when the phenotypes start to show through. Other people have transfected these tools into cardiomyocytes and found that you can use them, for example, to monitor cardiotoxicity. This here is a time course over 2 days of the action of pentamidine, which modulates hERG trafficking, I believe, in a negative fashion. Maybe. Not 100% sure. Anyway, it causes a cardiotoxicity, that's known, and here you can see the misregulation of the cardiac action potential waveform. And then this is some stuff that I'm pretty excited about, which is, I made the movie last night. This is work with Bridget Wagner and Amadeo Viteria in-house. These are INS1E model beta cells. I'm going to play you the movie. And you can see that the tool, at least nominally, works just fine in the cell line. And we can start to measure electrical activity in beta cells.
This is at baseline?
This is with high glucose. Oh. Baseline is quiet. So the reason I suspect my knowledge of beta cell electrophysiology is still nascent, but I see a lot of baseline wiggles under high glucose. And this was in glucose for like 20 minutes at this point. And then I see very sharp action potential sporadically. So we should talk about this more, actually. I know you have stories to tell me. And then this is another collaboration with Paula Barrett and Mark Binhacker at the University of Virginia. They study the aldosterone-secreting cells in the adrenal cortex. I embarrassingly didn't realize that the adrenal cortex wasn't even in the brain. Just so you know who you're dealing with. But despite my ignorance, the Optopatch was able to report action potentials in these cells as well. So now we're going to start asking questions about these cells are arranged in rosettes that release aldosterone. And the question is, they're gap junction coupled, but they don't seem to be firing together. So what's going on? What are the regulatory mechanisms there? These are stimulated with angiotensin to get them to go, but how does that mechanism work? These are all exciting questions that we'd like to pursue in the future. And then because I had 2 points, which was enough to draw a line, I realized that there are a wide array of hormone-secreting cells throughout the body, and at least from a preliminary reading of the literature, suggests that electrophysiology in a lot of these cells is either extremely nascent or not done at all, but the evidence that is out there suggests that most of these cells are strongly polarized, so that means that they could have potentially interesting electrical activities, and I'd love to start taking these tools around the body and looking for effects. Now, that's the pitch. Unfortunately, let me be totally fair and honest. Your typical day when you're using one of these tools, 9 o'clock, if you're good, you power up the microscopes and you begin the pre-check. 10 o'clock, you find out that for no apparent reason, one of your lenses is misaligned and you start fixing that. You get that done around 11 and you do sample prep. Then you get lunch and you start measurements around 1. Right, yeah, everybody lives this life, right? But we're supposed to be doing, it's supposed to be more high throughput than electrophysiology. Right, so then around 3 o'clock, the control code you wrote to run this software throws a bug, and you kick yourself because this is the bug that you knew you were going to fix one day, but today isn't that day. And then at 5 o'clock, you figured out the bug, and you're faced with a choice, the typical grad student choice, which is either you keep measuring and you're sad because you're there after 5, or you go home and you're sad because you didn't get the measurements you needed. And that's a little cartoonish, but this is approximately real data that it takes a new student in the Cohen lab about 6 months to build their first microscope. It takes an external collaborator between 12 and infinity months to get it done. Whereas somebody with experience who's done this before, you can get a microscope built. I've done it in 2 days, but that was my 7th one. So it can take less than a month as long as you're very familiar with the technique. And this is generally true of most advanced imaging methods. And so when I left Adam's lab, I really wanted to do something about the gap in application of these tools. And our solution, as we're pitching it right now, and as we're actually going to do as of 2 weeks ago, we got the official approval, is to set up an optical profiling platform at the Broad. Our goal is to bring techniques that are currently stuck in people's basements to biologists to run at scale and to make them robust. There are an array of imaging questions that can be answered by this. We already discussed optical electrophysiology and the potential it has for cardiotoxicity and for neuronal modeling and possibly for ion channel studies as well, if you guys want to talk to me later. But then there's also many questions about advanced screening, what genes affect cell morphology, protein function, translocation, biophysical parameters, all of these things, and of course, what drugs. And then there's a huge interest in mapping out the spatial organization of tissues as part of the Human Cell Atlas, the Human Tumor Atlas Network, and the Brain Initiative. And just like for optical electrophysiology, the tools exist. There are many in situ barcoding methods, such as those developed by the Blaney Lab. And there are an array of cell profiling tools, cell paint, of course, from the Broad and from Ann Carpenter. But of course, there's many other dyes, ROS detectors, metal ion detectors. There are many other optogenetic tools that we haven't discussed today for controlling protein function, for controlling gene expression. And there's good old standards like GFP fusions. At the same time, for spatial organization, the palette of techniques grows every day. But basically, there are ways to do multiplexed RNA hybridization. These are MERFISH, OSM fish, seq fish, star map, depending on how you count it. There's in situ sequencing to look for native variations in the mRNA. And there are tools for multiplexed antibody detection. And I should say that at this point, almost everything on this slide has been funded to go ahead within the platform. We haven't built any of it, but at least we have the approval. The techniques are real and generate data such as this. I won't belabor the point, but the point is that the tools as they were originally implemented are not ready for biologists to use routinely. On the left is the MIBI, the imaging mass spectrometer. In the middle is I found this sort of funny, this was one of Xiaowei Zhuang's former students, has now started his own lab, and this is very proudly showing how complicated the Merfish setup is. And I get it. I live the same life because over on the right is the microscope that I built to do Optopatch. And although it's great for a publicity photo, it's not great for actually running every day. So we want to replace those tools with things that are a little more refined, a little more robust. And we want to start interfacing these with the various groups and interest parties around the broad. So people who are interested in cell atlases and cancer. Neuroscience, of course, hits all three of these. Cardiovascular disease, diabetes, and of course, whatever else you may be interested in. Short term, we expect the platform will provide supervised, this is important, supervised access to the best optics available. We'll carry out complicated imaging experiments at scale, and we'll develop pre-processing data pipelines. This is important because a lot of the imaging generates data at an extremely high flux, and frequently you have a data set and you don't know what to do with it for months and years at a time. So we hope to address at least some of those concerns with the techniques we're developing. Medium term, we hope to be able to combine these tools into single pipelines so that you can look at both the electrophysiological profile of a cell, but also look at its transcriptomic profile or its protein expression complement, something like that. And long term, we want to optimize and create new experimental protocols, and we want to continue to onboard new techniques. Our hope is that perhaps some of these tools will become commercially available, and anybody will be able to buy a box cheaply and use it. And at this point, we'll probably shutter that part of the platform and move on to the next thing that's been invented by the optics community. Our idea is that here the platform members are in black and the user, possibly you, is in blue. The idea is if you have a new protocol, you have somebody to go to discuss what's possible optically, and there's someone who can help you. You prep a sample, we'll help you with the measurements, we'll help you with getting the right configuration. And then when you have an established protocol worked out, you can prep your samples at scale. We can do a little bit of secondary sample prep, and then we'll handle the bulk of the imaging, upload the data to cloud, pass it through a preliminary analysis pipeline, and you can hopefully download the result from your bed when the weather is like this outside. All of this, of course, will have to be supported by an interdisciplinary team of computational biologists, RAs, and optics experts who will be behind the scenes. And that's roughly where I want to leave you today. And most importantly, I want to ask for your input. What measurements would you like to do? We're still early days, so meaning I'm having the conversation about space in 15 minutes. So if there's something that I've forgotten or something that you would like to see, I'd really like to know about it. Just most importantly, I have about 100 people to thank. Most importantly, obviously, the work at the Cohen Lab. I need to thank Adam, who was an incredible mentor, both during my PhD and in terms of helping me get started here. I need to thank Aviv, who's my current boss, who really took a gamble on somebody to build something like this. And I've highlighted many of the people in Italics who have helped or have done work or have provided a lot of advice. But I specifically want to point out Carrie Wager, Tyler Brown, and Claudine Kohik, who helped me get started and navigate the Broad administration as I was setting this up. And that's it. Thanks a lot.